Good morning, everyone. Today's session is on our Phragmites Symposium. It's one of two, and it's our current approaches to management. And we've got a series of 5 talks today. The first talk is going to be from Samantha Tank on Great Lakes Phragmites Collaborative working towards a common agenda.
Great, thanks, Jason. Let me just share my screen quickly. Okay. Great, thanks so much, everyone. I am really excited to be here today, and I'm excited for our lineup of speakers. Samantha Tank, and I work as an aquatic invasive species program specialist at the Great Lakes Commission. So one of my roles at the Commission is to help coordinate the Great Lakes Phragmites Collaborative, and I'm really pleased that we're able to sponsor UMISC this year and host a special session. Especially pleased since we were able to still do this even though the conference is remote, but I think this week is going really well, which makes me even more excited that we have so many people registered and attending and are able to see all of these great talks. So I'm going to kick off the session with some background on Phragmites, as well as an overview of the Great Lakes Phragmites Collaborative. I'd first like to acknowledge my co-authors, so Erica Jensen and Patrick Caniff from the Great Lakes Commission, and then Kurt Kowalski from USGS. I'd also like to give a special shout out to Tasha Tucker from USGS as well. So sometimes referred to as common reed, Phragmites is an aggressive wetland invader. I often like to talk about it with people who haven't seen it before, or don't think they've seen it before, or it's new to them, as it's the invasive species that once you see it once and realize what it is, you're gonna see it everywhere. You can see from these photos, it's not that uncommon to see mature stands grow 4+ meters tall. And the stands form these really dense, thick near monocultures where the dead biomass that dies off each year can remain standing for several years, which further shades out native plant species. I've been in several mature stands where you go in, you're like maybe a couple feet into the stand and you're completely surrounded by Phragmites, and it seems like it's dusk even on a sunny day. Phragmites arrived in the eastern United States in the late 1700s, early 1800s from Europe. So it's been here for quite a while and was likely incidentally brought over that many years ago as ship ballast material, but then was also likely intentionally brought over in later years. One of my favorite stories that I can't speak to its truth, but I think it's kind of a fun story, is that Phragmites was brought over as elephant bedding for elephants traveling along the surface. So not sure if it's true, but as you can see from this map by Saltston et al., Phragmites is not just this localized issue or something that we're dealing with here in the Midwest, but it's this binational problem across North America. And when I think about the impacts of Phragmites and what it has on the environment and on people, I tend to think about them in 3 different ways. How Phragmites is impacting our water, our wildlife, and then us as people, our way of life. So these dense stands, which I said are near monocultures, they can alter flow, they can clog drainage ditches or ponds, and they can even reduce water levels. Their density can easily outcompete native wetland plants as well for space and light. Phragmites is known to impact a multitude of wildlife species as well. Large patches of Phragmites don't provide suitable habitat for marsh nesting bird species and even provide fewer insects for foraging birds than a native wetland would. Phragmites stands also eliminate suitable habitat for threatened and endangered species, such as reptiles and amphibians. And there's lots of organizations that are working to manage Phragmites for fish and wildlife habitat restoration, such as the U.S. Fish and Wildlife Service, Ducks Unlimited, Nature Conservancy. But beyond just disrupting our ecosystem services and the plants and animals that would take up these habitats, Phragmites stands also directly impact people as well. And this is something that is meaningful when you're talking about invasive species, especially to the public. These stands can obstruct access to recreation areas that are used for hunting, fishing, swimming, or hiking. And when people walk through them and they get the seeds on them or they have fragments, they can actually continue to spread Phragmites, and it becomes even more of a problem. There was an article that came out not too many years ago that showed that Phragmites on your property can actually decrease the value of that property. So that's obviously a concern for landowners. Excuse me. And Phragmites can even pose a safety threat by blocking road signs or creating a fire hazard. So this is a really large-scale problem that needs a large-scale solution. So this is why in 2012, partners from the Great Lakes Commission and USGS came together to establish the Great Lakes Phragmites Collaborative. The collaborative was formed to facilitate communication among stakeholders. And we do this through our mission. So the collaborative's mission is to facilitate communication among stakeholders across the region and serve as this resource center for all things Phragmites biology, management, and research. So while the collaborative administrative staff might not be out on the ground managing Phragmites or conducting research, although I will say some of us are actually doing those things, you can believe that the entire collaborative community is. So the collaborative is much more than us at those two organizations. Helping to guide the collaborative is this advisory committee that represents a binational cross-section of Phragmites experts. So the advisory committee, you can see their member organizations on the screen. They are providing guidance and leadership to the collaborative as it works to develop products, facilitate communication, and foster collaboration across the entire region. But beyond just our groups of agencies, the Phragmites community is also a part of the collaborative. So this includes anyone who's managing Phragmites, conducting research, trying to positively impact the ecosystem by removing Phragmites, or being impacted by non-native Phragmites. So that means that everyone on the line listening right now is part of the Great Lakes Phragmites Collaborative. So I want to talk a little bit about the approach that we use. The collaborative uses this approach called collective impact to guide the work that we do. In 2016, the core team published a paper about how we use the principles of a collective impact to manage the Phragmites Collaborative. If anyone hasn't read that and is interested, just shoot me an email and I'm happy to send it your way. So there's 5 conditions of collective impact. There's a neutral backbone organization, and we at the commission serve as the neutral backbone. And what we do is we anchor the group work and provide long-term administrative support. The collaborative also maintains open lines of communication with a broad range of stakeholders across the region. And we do this through maintaining our listserv base, maintaining our website, hosting webinars, and then we have other communication products that we have put out over the past nearly 10 years. So over the past 4 years or so, the advisory committee with the core team has been developing a common agenda. So this is an essential part of the collective impact approach, which is defining our shared path forward through these set goals and objectives. I'm going to talk about that more in just a minute. We're also working to establish a shared system of measures to evaluate the effectiveness and progress of our common agenda and the member strategies that are included within it. And finally, we're working to promote mutually reinforcing activities among our members that will align efforts towards achieving our collective goals. So utilizing this collective impact approach really provides us with a systematic way to get from all of the problems that are currently surrounding us in regards to Phragmites management and research, such as funding gaps or uncertainty about best management practices or communication barriers, and then get us to our vision of a Great Lakes ecosystem that's not invaded by non-native Phragmites. So just going back to the common agenda, this is a big first step that we can take to align our shared goals and establish a shared vision for change. So the purpose of the common agenda is to clearly define the intentions of the collaborative and then develop a shared understanding of gaps and problem areas surrounding Phragmites so that we can highlight the areas and strategies that we're working on or that we wish to work on in the future. So we have a news release that's going out today, actually. So I'm really excited that we can share the final product with you right now. You should see that coming through if you're subscribed to Glenn announced through the Great Lakes Commission, or I'll be sending it out shortly through our listserv. And so we're just finalizing and starting to promote the common agenda. This is what it looks like. So it's broken up into 4 focus areas, and those are regional coordination and collaboration, best management and restoration planning, the advancement of research and technology, and funding for Phragmites management. So each of these focus areas has a subset of member strategies. You can see that there's 3 or 4. So I'll just go to one and we can look at this a little more closely. So the advancement of research and technology focus area, there's 3 member strategies. There's supporting and implementing the Phragmites Adaptive Management Framework, or PAMF. And I'm actually going to kick off the second session talking about that. Providing a forum for researchers to collaborate and communicate with managers, which is really what we're doing right now. And tackling and sharing the latest advancements in research and technology throughout the wider Phragmites community. So here's some of the things that the collaborative, some of our products that the collaborative has put out over the past 10 years that is supporting the charge of the common agenda. We have developed a lot of different resources such as our website, which if you haven't checked it out, here's the link at the bottom of the screen. I'd really encourage you to go and browse around. We have our webinar series where we have 39 archived webinars at this point, and they focus on all different things. There's research that we send out quarterly through the listserv. We're active on social media, and we host an invasive species organization map as well. So these are just some of the things that we've been working on over the last 10 years. I didn't want to leave you without things that you can do to support the common agenda. So some of the things you can do, well, you're already doing it. You're here, you're engaging with the Phragmites community. So thanks for joining this session again today. But you can also share relevant Phragmites management research and resources with the community. And you can do that by sending them directly to me. My contact information will be on the next slide, or you can post through our listserv as well, and we can help you get that information out there. You can also engage with others and work collectively to manage Phragmites. So remember when I showed that map in the beginning, Phragmites isn't this isolated issue, and so we don't want to work alone. We want to work together collectively to solve it. And then you can also consider how your current activities could be adjusted to align with our member strategies that are laid out in the common agenda. And finally, you can join PAMF, and you'll have to stick around to hear more about PAMF in the next session. So with that, I'd like to just acknowledge the Great Lakes Restoration Initiative for providing funding. And thanks, everyone. I'll take any questions if there's time.
Yeah, thank you, Samantha. There's plenty of time. You've got about 6.5 minutes left to go over any kinds of questions. From our other moderator, there's not any questions right now from the audience, but I've got a few of them. So one of them would be is, what if you're a private landowner and you're dealing with Phragmites on your property at a very small scale? How can a smaller landowner get involved in PAMF and then follow the guidance that you're providing for, say, larger organizations on statewide or county levels?
Yeah, and so just to give a little bit of a background on PAMF to make that question a little bit more clear to those who aren't familiar with it. So this is a basin-wide program where anyone from private landowners all the way up to federal agencies can enroll, and they can tell us a little bit of information that we give them ways to monitor about their Phragmites infestation, and then we provide them with best practice guidance that's supposed to be a nice balance of effective and efficient. So landowners are actually one group that we really love engaging with. This is a free program. Anyone in the Great Lakes region can participate. So all they have to do is reach out to us, and we give them a training, which we've actually migrated all of our training materials online since the pandemic began. And so they can take these online trainings, which only take about 2 hours. We'll send them a monitoring kit and show them how to use it. And then we're available for remote support as well. So they just need to follow our monitoring steps, and then we'll give them information on what's likely going to most effectively and efficiently take care of their Phragmites problem. And then the collaborative website has lots of tips and tricks for identification, or we have a contractor database that it's under construction, but it will be back up soon. We have the SYSMA contacts so that people can reach out to their SYSMA coordinators and get help and advice. So lots of things on the website for private landowners as well. So we're not just targeting experts, we're trying to reach everyone. And private landowners are one of those groups that can be tricky to reach, but we really enjoy engaging with them and see a lot of impact.
Thanks. Another question would be about the common agenda. Could you refer back to one of your earlier slides? It has like the larger table in it. So with this, and you're putting forth the common agenda as a draft, how could our audience, how can they take this and then make comments that are going to be most useful for the advancement of the common agenda?
Yeah, so this is our final product at this time. So what we want everyone to do is take a look at this and see what work that you're doing currently that is aligned with the common agenda. Or if you're doing work that, so say you're managing Phragmites, for example, but you aren't really connecting with others in your area, we want you to then look at our member strategies, see, oh, well, one of the member strategies is working on a regional scale, right? And helping to advance Phragmites management by collaborating with others in the area. So I'm going to go and I'm going to do that, and I'm going to broaden my research, or I'm going to broaden my my management as best I can and collaborate with others. So we're really just putting these out there so we can help the work that's already happening be more strategic, be more collaborative, and have a bigger impact overall. Because at this time, we have a limited set of resources, and we'll always have a limited set of resources. But when we work together, we can expand those resources and we can make a greater impact.
That's great. You've got about 2 more minutes. So if there's any other questions from our audience, please put those in chat. Or Samantha, if you want to, if there's any other details that you'd like to go into further about, you've got about 2 minutes.
Yeah, no, this is great. I will say again, just go ahead and check out, I'll go back to my final slide. Go ahead and check out our website if you haven't already. And thanks again to all of the speakers who have agreed to present as part of our Phragmites session. I hope I've set you up well. I tried to give a bit of background so that if anyone watching is new to Phragmites and isn't aware of the impacts of the species, you have a little bit more information now when we get into all of these really interesting topics that we have coming up.
Okay, great, thank you. So Travis, if you'd share your screen, please, we'll get you set up for the next talk.
Okay. Is it going to work? Can you hear me all right?
Yep, I can see and hear you just fine.
Okay.
So we'll just have a minute before you get started. That way we're all consistent on time. Okay, our next speaker is Travis Bartnick from the Great Lakes Indian Fish and Wildlife Commission, and his topic today is non-native Phragmites management in coastal southwestern Lake Superior.
All right, thank you very much for that introduction. My name is Travis Bartnick. I'm a wildlife biologist with the Great Lakes Indian Fish and Wildlife Commission, and I've been overseeing GLFWC's invasive species program since late 2016. So thanks for being here. I'm going to be discussing some of the non-native Phragmites efforts that have been underway in the southwestern portion of the Lake Superior Basin in recent years. So this presentation will be focused on two areas in the southwestern portion of the Lake Superior Basin, the Chequamegon Bay area and the St. Louis River estuary of Duluth Superior. First, I'd like to share a little bit about the organization that I work for. So GLFWC is an intertribal natural resources agency that works with 11 Ojibwe tribes who exercise treaty rights in portions of Minnesota, Wisconsin, and Michigan. We assist with the implementation of off-reservation treaty rights and do so under this framework of co-management with other government agencies. So co-management is the concept where multiple entities such as tribal nations, state and federal agencies have this shared responsibility for the stewardship of our natural resources. And so here we have a map showing the distribution of the 11 Ojibwe tribes that I work for in the ceded territories, and to give an idea of how large of an area that we're working in. I'd also say that the majority of our work is conducted in the 1837 and 1842 ceded territories you can see on the map there. And so part of GLFWC's mission is to protect ecosystems in recognition that fish, wildlife, and wild plants cannot long survive in abundance in an environment that's been degraded. And so the degradation of native ecosystems by invasive species or non-local beings poses a serious threat to the continued exercise of these treaty reserved rights and the traditional lifeways that they sustain. And so you heard me say this term, non-local beings. And so some in the Ojibwe community take issue with the negative connotation with the term invasive species and have suggested using an alternative that roughly translates to non-local beings. So you might hear me use that term occasionally in place of invasive species. So GLFWC's invasive species program started in the late 1980s with focus on lamprey and purple loosestrife. And over the years, the program's developed into a more comprehensive program focusing on a wide variety of terrestrial and aquatic invasive species. The program consists of 5 components, including prevention, involving education and outreach, early detection and rapid response, control and management, research, and cooperation and coordination. So many of us are familiar with non-native Phragmites, and Samantha gave a good overview in her presentation. But I think the key points here is that the species can form these dense monocultures, can reproduce with seeds, rhizomes, and overland runners. We also know that floating root fragments can aid in the dispersal of this species, and that human activities have been a big driver in the spread of this species. GLFWC has prioritized the prevention and control of non-native Phragmites because of the impacts on native ecosystems, especially with concerns of how it may impact treaty resources, such as wild rice, and restrict access to areas for cultural uses. GLFWC's work with Phragmites started in 1999, and the tracking of occurrences and treatment efforts started in 2013. So Chequamegon Bay is located about 60 miles east of Duluth Superior, and the first populations around the Chequamegon Bay area were discovered within a mile of 3 wastewater treatment plants in the area. The wastewater treatment plants established non-native Phragmites reed beds to aid in their sludge dewatering efforts. And despite early statements by contractors saying that they wouldn't spread by seed, Genetic studies conducted by Red Cliff Band of Ojibwe and others suggested that they were in fact spreading by seed from these wastewater treatment plants. So our control work has been conducted typically in late July through August each year, mainly using herbicide control with glyphosate and imazapyr. And just in the last couple of years, Glyphwic has phased out the use of glyphosate, so we're primarily using imazapyr on these sites. So here's an example of one of the wastewater treatment reed beds using non-native Phragmites in their sludge dewatering process. 3 of these reed beds were installed in the Chequamegon Bay area in the late 1990s and early 2000s. And by 2013, many sites were documented within 1 mile of these reed beds. We also suspect that the local ATV trails that run by these wastewater treatment plants, as well as equipment used at the reed bed sites, were also potential vectors of spread. This map shows some of the roadside, lakeshore, and other survey efforts conducted by Glyphwic staff over the years. As you can see, most of these efforts have been focused within the Lake Superior Basin in northern Wisconsin and the western portion of the Upper Peninsula of Michigan. And here I've included just a couple of other maps from our annual reports showing some of our Phragmites survey and control efforts. The Phragmites surveys are indicated by the yellow lines. And then the purple areas, which might be kind of hard to see in these, but those are indicating all of the monument or wild rice water bodies in the region. And then just one more map kind of showing some of these efforts over the years. And here we have satellite imagery of the Chequamegon Bay area with the red dots indicating non-native Phragmites locations, and then the green dots are showing native Phragmites sites. And so we have a lot of native Phragmites in the region, and it's really important. We've learned that it's really important to know the difference between the two, just so we can really focus our efforts on those non-native Phragmites sites, and so that we're not accidentally spraying herbicide on the native species. So here are some photos of what a few of the sites looked like in 2013, right after they were first discovered. They're not super huge, but they're all typically maybe a quarter of an acre or so or less in size. But they were getting to the point where if something wasn't done, they were going to continue to spread rapidly. And after a few years of effort, this is what a lot of the sites are starting to look like now. If they haven't been completely eradicated, what we're seeing are these sites with sparse shoots coming up, usually pretty stunted, typically less than 50 shoots per site now. And so we're seeing a lot of progress at these sites after continuing for several years in a row now. So in 2018, the Red Cliff Band of Lake Superior Ojibwe and partners undertook a huge effort to eradicate the non-native Phragmites from these reed beds at the wastewater treatment plants around the Chequamegon Bay area. And they replaced them with native Phragmites, locally sourced native Phragmites. And so this effort's been described in other presentations, so I'm not going to go into super detail on everything that occurred with this project. But I think it's worth just pointing out that this has been a huge effort. And a lot of cooperation, coordination, and funding went into this, and it's going to be extremely important for in the future for controlling the species going forward. And so despite all the successes that we've experienced so far, there's still a lot of work to do. New sites do keep getting reported each year, and we're rapidly responding to any new sites as well as monitoring existing known sites. These photos show Phragmites growing at a residence located within about 1/8 of a mile of a wastewater treatment plant. And this site was reported and controlled with herbicide in 2019, and no new growth was observed at this site in 2020. I also want to stress the importance of annual follow-up monitoring at these sites. So this is a photo of a site in the city of Washburn in 2013. The site was last treated with herbicide in 2014, and no new growth was observed during follow-up monitoring all the way up until just this year in 2020, all of a sudden we noticed a few new shoots growing up, and we suspect that this probably has to do with the mowing operations conducted by the city in this right of way. As you can see in 2013, it was mowed all around it, and after 2014, they were probably mowing some of these smaller shoots that have been coming up each year, and our crew, our control crew was probably just missing some of these shoots because they had been mowed in those subsequent years after the last herbicide treatment. So just, showing how important follow-up monitoring is even several years after you think you might have eradicated it from a site. And so that does it for the focus on Chequamegon Bay. Now I want to shift to the St. Louis River estuary. So in 2014, Glyphwick conducted shoreline surveys and documented approximately 70 occurrences over an area combined of less than 10 acres on both, combining both the Minnesota and the Wisconsin sides. So Glyphwick and the DNR and other partners planned a response to these observations in late 2014. The initial treatments occurred on the Wisconsin side in 2015. And this ended up leading to the establishment of the St. Louis River Estuary Phragmites Technical Team, which had partners on both the Wisconsin and Minnesota side of the estuary. So Glyphwick conducted control work, follow-up monitoring, and additional surveys on the Wisconsin side in the following years after 2015, and then eventually worked with the Duluth Stream Corps through Community Action Duluth in 2017 to start treatments on the Minnesota side. Glyphwick has continued monitoring and treatment efforts on the Wisconsin side, and the Duluth Stream Corps has been continuing those efforts on the Minnesota side ever since. And so this map on the right is just showing some of the results of those initial surveys in 2014. So as of 2018, about 3 years into the control efforts, about half of the known sites on the Wisconsin side had been eradicated. And then now in 2020, the number of sites on the Wisconsin side have been reduced to about, I think, 3 or 4 sites. Unfortunately, we weren't able to get out there as we had planned this year because of the coronavirus. So that kind of thwarted some of our plans. So we did work with a partner to get out to some of these sites just to do some follow-up monitoring for us this year. But we are planning on a more extensive, comprehensive survey of the shoreline, hopefully next year. Sites on the Minnesota side have also drastically been reduced thanks to the efforts of the Duluth Stream Corps. They had much larger populations to start with, and they've been using a control approach of conducting a winter knockdown, followed up by herbicide application in late July and into August each year. So one of the biggest challenges has been working with some of the landowners in this area, because on the Minnesota side, the Duluth side, it's heavily industrial. There's a lot of railroad in holdings, and so gaining access to some of those properties has proven difficult. Here I just have a screenshot of Glyphwick's interactive Phragmites map. So we have verified native and non-native Phragmites occurrences throughout the region. So if you're interested in that, you can take a look at that map. So with the efforts of many in this region, we've seen a lot of successes in preventing the further spread or reducing populations of non-native Phragmites in the coastal area of southwestern Lake Superior. We've helped educate tribal youth, tribal harvesters, and the public of the importance of preventing the spread of this invasive species or non-local being. Our work is aimed at protecting treaty resources and ecosystems under Glyphwick's mission. And so we've developed great working relationships with many partners in these efforts. And so far we've avoided having to take more drastic measures, as we've seen in areas where the species has grown beyond, seemingly beyond control and eradication. We still have many challenges associated with this work, whether it's securing a reliable source of funding, dealing with turnover at some of our partner agencies or organizations, data sharing logistics. There's a continuing concern with the use of herbicides. Landowners are still unwilling to cooperate in some areas. And then we've noticed commercial marketing using non-native Phragmites seed heads, as you can see in that photo, the rooster there. And then there is continued use of reeds for duck hunting blinds. And so we really want to increase those outreach efforts to duck or waterfowl hunters. And so this work would not be possible without the effort of many partners in the region, and this has helped us build many important relationships that I hope that we can continue into the future. A lot of the work that I've talked about was conducted using funding from the Bureau of Indian Affairs, the U.S. Fish and Wildlife Service, and the EPA. And with that, I can take any questions or comments.
Great, thank you. You've got about 5 minutes for questions. So we've got two from the audience. One is, are you seeing any residual effects from a mesosphere that negatively impact the restoration of plant communities?
I think they've been seeing a little bit more of that with some of the larger sites on the Minnesota side of the St. Louis River estuary. In terms of around the Chequamegon Bay area, you know, there's been a little bit of a residual effect, but we're really careful with our control crew to be very conservative with the use of herbicides. And, you know, in sensitive areas, we do have them use hand swiping when possible, just to avoid, you know, over application with the sprayers and that kind of thing. But yeah, I think that is a concern that we're going to have to discuss with some of the sites on the Minnesota side that had much larger non-native Phragmites patches.
Second question is it difficult to identify and control non-native populations that emerge within native populations?
It's more time consuming, and you really have to have a good eye for that identification. If they are growing kind of interspersed or next to the native species, you know, sometimes what we'll do is we'll bundle the ones that are identified as the non-native and then hand swipe those.
For your restoration sites, do you have any plans to do any reseeding of those areas?
That's something that we still need to, I guess, work into our management, our stewardship efforts. You know, we've really just been focused on, you know, we see this as an opportunity. The sites are so small that we feel that they're still at the point of eradication. We do realize that, you know, we have to have a plan going forward for the restoration of those sites. But it's something that we have to discuss further within our Phragmites technical team.
Someone asked, can you give additional details about your burning protocol for handling Phragmites?
I'm sorry, with our what protocol?
Your burning protocol. Do you burn any sites?
We do not burn any sites. No, I think maybe they're referring to, yeah, so I guess I had this photo of the burning here, which was, I think, over in the Green Bay area. And this is what I was pointing out that we've been fortunate enough to avoid so far, was having to result to these large scale burns that they've been doing where the non-native Phragmites has really gotten out of control.
Another question is there a precedent for use of native Phragmites for wastewater treatment management in the area?
I think the Red Cliff project is probably one of the first, I guess, projects that has done that, to my knowledge. Somebody, Julia, who's going to be speaking, might have a little more information about that. But I think this is almost kind of seen as a pilot or an experimental project. And to tell you the truth, I don't know how those native Phragmites that were transplanted into those wastewater treatment plant reed beds, I don't know how they've been doing so far. So I guess I'd have to follow up with some of the folks that are involved with that project.
And the last question is, would you please share the link for the native and invasive Phragmites map again?
Oh,
and
sure.
how the public can get access to it?
Yeah, so I think the easiest way to, you know, find this is just in your internet search engine. If you could do Glyphwick Phragmites. But I do have the link down below here, and I believe that this presentation will be shared in the future.
Great, thank you.
Right,
so we've got about a minute and a half left for any additional questions. Okay, our next talk
All right.
is by Julia Bowen, Coordinated Response to Invasive Phragmites in Minnesota.
All right, do you see my presentation and can you hear me?
Yes, I can.
Great. I'm Julia Bonin. I'm a researcher at the University of Minnesota and have been working closely with Dan Larkin and Sue Galadowicz from the University of Minnesota on our MinFrag program, which was the initial research that we started with in invasive Phragmites, and also working closely with Wendy Kroll from the Minnesota DNR. We have been working for a few years now. Actually, first I'd like to just thank the Great Lakes Phragmites Collaborative for the opportunity to provide an update on some of the work that we've been doing in Minnesota to address non-native Phragmites. As I said, it's been a very productive collaboration with different state agencies, including the Minnesota DNR, the Minnesota Department of Agriculture, the Minnesota Pollution Control Agency. And then I would note that our effort relies heavily on the contributions of county and local government entities, as well as a lot of private citizens who have been engaged. There we go. Our initial Phragmites research effort at the U of Minnesota, U of M, was dubbed MinFrag, and it was initially funded through the Minnesota Aquatic Invasive Species Research Center with the goal of filling the knowledge gaps in our understanding of three key pieces of information regarding invasive Phragmites in Minnesota. First, we needed to know how widely distributed invasive Phragmites was. And then we asked, how great is the risk that it might be spreading from seed in Minnesota? And finally, we wanted to know, how can we effectively respond to invasive Phragmites to control it? Our research was prompted in part by a desire to change the regulatory status of European common reed from a restricted noxious weed to a prohibited controlled noxious weed. And the first effort to do this failed when it was determined that there wasn't enough information about the distribution of invasive reed in Minnesota to make that regulatory change. So the first task of this research was to document the distribution of invasive Phragmites in the state. There was also a concern that we didn't understand the risk of spread by seed. And so to fill that knowledge gap, we sampled populations to assess their genetic diversity and seed viability. And I'll be talking about this a little bit more. And the last piece of our initial research was to assess strategies to control invasive Phragmites. So our research that we conducted from 2017 through 2019, and with that research, we've filled some of those knowledge gaps. To date, we've documented 533 verified populations of invasive Phragmites in Minnesota. I would note that that's more than 100 since I last gave this talk, 100 additional populations since I last gave this talk to UMISC in 2018. And those occur in 40 out of Minnesota's 87 counties. So you can see their distribution on the map. And you can see that there's high regional variability in where those populations occur, as well as the density or the number of those populations that occur in each region. In addition to invasive Phragmites in the landscape, in Minnesota, we have 15 wastewater treatments that have used or continue to use invasive Phragmites in reed beds to dewater biosolids. I would note that 3 of the facilities sprayed out their own reeds. 2 of them did that just this fall, and 1 of them did it several years ago. And the reeds in the 4th facility that don't have it any longer, those just spontaneously died out a few years ago. So we know that patch size, or the density of populations in an area, confers greater potential for genetic diversity and subsequently greater potential to develop viable seed. In Minnesota, as you can see, most of our populations are still relatively small, and many of them, again, are isolated in the landscape. So the small patch sizes and the low density means that we're likely to have lower genetic diversity and thus low seed set. In addition, the small patch size means that the majority of the populations are still going to be quite manageable, both in terms of the complexity, the types of equipment you have to get out there to access and treat those populations, and then consequently the cost of treating those populations. So that's the good news in Minnesota. To assess the risk of spread by seed, we divided Minnesota into 9 regions that account for the north-south temperature variation and an east-west moisture gradient. And we collected leaf tissue for genetic analysis from about 52 populations, and we collected seed heads from those same populations for a seed viability assessment. And based on our assessment, we found that viable seed did not develop in the northern 1/3 of Minnesota, but it did develop in the southern 2/3 of Minnesota, as you can see from our map here. And you can see also from the chart that not all of the populations that we tested, even in the southern part of the state, developed seed. So there are populations down there that are perhaps just not genetically diverse enough, or they're not in dense areas with diversity in the region, in the area, so that they did not develop viable seed. So we repeated the seed viability study for the years 2017 and 2018, so seed collected in those years. For genetic diversity, note that although there's a difference in the magnitude between the years, the trend is the same. There's a positive relationship between genetic diversity and viable seed production. And likewise, there's a relationship between latitude and viable seed production. On the right, latitude 48, that represents our most northerly populations in Lake of the Woods County and in St. Louis County, so on Highway 11 and Highway 53 up near Orr. Those had no seed development. And then latitude 43 on the left is represented by a population from Mabel in Fillmore County, which is down near the southern border of Minnesota. And so seed does develop at those lower latitudes, and the viability of the seed at those latitudes does vary, but in general, the seed index follows that latitudinal gradient. So the third part of our MinFrag research project was to create a framework for organizing a response to invasive Phragmites in Minnesota, so treating it. Chelsea Blanke, who's now with the Minnesota DNR, pulled together all of our occurrence data and information from contractors in terms of costs, and generated a really comprehensive assessment that provides guidance for this response phase of our program. The assessment is developed around a regional framework that relies on local expertise from each of those regions. And the document assesses control approaches based on the conditions and size of each particular population. We had enough information about each of them from having visited many of them that we could make pretty specific guidance for each of those regions. And it then provides the cost estimates for 3 years of herbicide control and mowing treatments. And that was estimated to be between just over $800,000 and $2 million. So the framework that was provided in the response assessment document has been helpful for considering how to approach the control phase of invasive Phragmites in Minnesota. And it's being used as we begin to coordinate control efforts. We've already given some additional thought, I would say, to the response to invasive Phragmites, and realizing that we need to roll out treatment, monitoring, and surveillance in each region based on the needs of those particular areas. And so this map looks at the state through that slightly different lens and will help us focus on the different levels of effort needed in a way that matches how the response is taking shape in each of those areas. So I would note that I was supposed to give a talk to the annual meeting of the county agricultural inspectors this summer. And of course that was canceled due to COVID. But I hope to find another opportunity to address that group, because I still haven't engaged all of them. But in the counties where we've documented Phragmites, they've been great local resources to help facilitate control. And in the areas of the light pink where only surveillance is needed, and we've not yet documented invasive Phragmites, we hope we can engage them to do the surveillance that'll be needed to ensure that if invasive Phragmites does establish in those counties, we can document it and treat it in a timely manner. So outreach has been a really important part of the response, and there's more outreach to be done for our response to invasive Phragmites in Minnesota. State and local agency staff and private citizens have been essential to the effort to document invasive Phragmites in the state. So that's the surveillance component of the response effort. But they have to be engaged first through outreach efforts. And likewise, as we move fully into the management phase of the response effort, coordination with these same stakeholders will be critical to the comprehensive control program. Monitoring and ongoing control will be key to the successful control effort. And as we know from what Travis was saying, that will be a long-term effort. So we will need some sustained support. And as we know, the goal of any response effort is preventing, stopping, reversing the spread of the invasive species. And here in Minnesota, we're optimistic that we're at a stage in this invasion where it's reasonable that we could stop the spread of invasive Phragmites and prevent future establishment. So here are some examples of efforts in Minnesota to control invasive Phragmites to date. And so Travis talked in detail about what's going on up in Duluth. So the Duluth Stream Corps, GLFWIC being engaged with Community Action Duluth since 2016. They're having great results, as he noted, on the populations they've been able to access. In the Chisago Lakes area and the White Bear, the Chicago Lindstrom Lakes Association and the White Bear Lake Conservation District have both been managing Phragmites on their shorelines for the last 2 years. And this effort has been backed by a significant contribution from volunteers from the different lakes associations. They've been working very hard in their area to protect their assets. Several of the metro counties coordinated efforts this fall, or they started that effort last year even, but they acquired some funding from the Department of Agriculture and then hired a contractor to do treatments this fall throughout the metro area. They collectively were able to treat over 50 different populations. I know they still have more work to do and they plan to continue that next year. And then the Minnesota DNR has acquired funding from the Great Lakes Restoration Initiative to conduct treatment. Unfortunately, we were not able to fully deploy those funds this fall, but we do have those funds available yet next year, and we'll continue to do treatment in outstate areas of Minnesota. The treatments this year that we did do were primarily in Candy-Ojai County, where we made a nice impact there. And Wendy Crowell and I will be planning this winter in preparation for next season. I think we perhaps underestimated how much effort it will take to actually plan and coordinate all those treatments. And we have some lessons to learn from others who have already been deploying their grant funds and doing restoration work. And then the Minnesota DOT and county highway departments have been doing some treatment, but more work needs to be done to coordinate efforts with them and to document the treatments that they've been doing in a central database, which for our purposes, we're using EDMAPS. And then there are certainly other efforts going on in other parts of the state as well, with different county ag inspectors doing some treatment, et cetera. So to date, treatment has occurred in at least 20 of the 40 counties with known populations of invasive Phragmites, and that accounts for more than 32% of the documented populations that have been treated at least once. And I'm not able to provide today a summary of the area that has been treated at this time. We're pushing to have all of the treatment records documented in EDMAPS so that we can track the control efforts on a state level. We had a long fall spray season this year, and so those records haven't all been updated at this time. But I'm sure I'll need to also push to track down some of those records and assist with getting them in EDMAPS, which we anticipate will be our central database for that. And then I would note that we're also working with Chuck and his staff at EDMAPS to make modifications to EDMAPS Pro so that we can efficiently use it for monitoring all of the treated populations. So in 2013, the Minnesota Department of Agriculture listed non-native Phragmites as a restricted noxious weed. And this designation prevents the transport and sale of propagating parts. But that regulatory status of non-native Phragmites is likely to change in the coming year. In December, the Noxious Weed Advisory Committee will vote to reclassify non-native Phragmites as a prohibited noxious weed, which would facilitate our control efforts because landowners would then be required to control it or prevent the spread of it on their property. And the Minnesota DNR is also in the process of rulemaking to list invasive Phragmites as an aquatic invasive species. And this double listing by both the Department of Ag and the DNR would be similar to the way purple loosestrife is currently listed in Minnesota. Invasive Phragmites in Minnesota is a complex situation due to its continued use in 11 of the wastewater treatment facilities in the state. And although some of the municipalities, as I mentioned, have already voluntarily taken steps to stop using invasive reed as part of their dewatering process, other municipalities have not been able to and will not be able to transition so quickly. As it stands, any facility that's currently using invasive Phragmites will be granted a transition period to develop a plan to move away from invasive Phragmites if the status of invasive Phragmites changes from restricted to prohibited, as we anticipate will happen. Dr. Dan Larkin at the University of Minnesota will be leading a research effort to provide information to assist the transition, and his research will address 4 particular needs to facilitate that transition. One research project will be to screen robust native populations as an alternative to using invasive Phragmites in the reed beds. It's likely that native reeds will not be as effective at dewatering as invasive reeds are, but we currently don't know what level of efficacy we could expect from the native reeds. And I would note, based on what the question that Travis got, that we would be promoting the use of local source native Phragmites, which is what happened up in northwestern Wisconsin, where 3 wastewater treatment facilities are currently using local source native reeds. And I don't know the status of their functioning right now, but currently we have 2 facilities in Minnesota that are using native reeds. One of them has been established for a while, and their reeds are operating pretty well mixed with cattails, invasive cattails. And the other facility is using a source of reeds from Nebraska. So we'd like to consider using invasive or native reeds from Minnesota. And then another research project will look at developing methods for propagating and establishing those native reeds so that the facilities can be more fully operational more quickly. So right now it's taking a little while to get those reeds established to a level that's satisfactory for functioning, for the wastewater treatment facility to function properly. And another angle of the research will assess cost-effective methods for eliminating non-native Phragmites from the reed beds. Based on our surveillance of land application sites, we believe we can safely clean out the beds using parameters very similar to those currently implemented for land application of biosolids from the reed beds. And that should save quite a bit of funding or costs for the communities that have to do this transition. And then finally, we'll be looking at developing best practices for maintaining invasive reeds in the reed beds during this transition period. So for example, can we implement some methods to prevent seed development and or seed dispersal in those facilities while they continue to use the invasive reeds? And we're already collaborating with the wastewater treatment facility operators, and we'll continue to do that because we require their expertise to come up with solutions that will work for them. So we anticipate working closely with them as we do each aspect of this research program we have coming up starting in January, and then again, some funding coming in June. And with that, if I have time for some questions, I'm happy to take them.
Yes, you've got 3 minutes for questions. One of the first ones is, some of the wastewater treatment facilities seem to have more prominent invasions surrounding them than others. Any insight into what might make the frag at one plant more likely to result in escapees than the others?
Well, one thing that we don't know from our facilities is whether or not they're producing viable seed. I assume that would be the primary reason that the Phragmites would be escaping from the facilities based on our, we've actually done a pretty good survey of all of our facilities in Minnesota to find out what practices they're using. And we don't think that it's likely that it's fragments of the plants escaping. So then it seems likely that it's seeds. And so, for example, we have one northern one that it's maybe too far north to have for viable seed to be able to develop. But alternatively, perhaps it's just not a genetically diverse enough population where it's likely that seed would develop. And so those are the 2 kind of reasons why we think maybe some are more prone to seed development and others not, and thus escape into the landscape.
And then the other question is, by what mechanisms do increased genetic diversity lead to increasingly viable Phragmites seeds?
That's a good horticulture question. So just having the genetic diversity in it, they're self-incompatible, I guess. That's the answer to that question. So you have to have cross-pollination for them to be able to develop seeds. So if you have a distinct genetic type next to another distinct type, then those 2 can cross-pollinate and produce viable seed.
Okay, that's great. Thank you. Thank you very much.
You're welcome.
We have our 4th speaker today, semi-automated identification and monitoring of Phragmites australis using drones and satellites by Connor Anderson.
right. So my name is Connor Anderson. I'm a PhD student at the University of Minnesota, part of the Remote Sensing and Geospatial Analysis Lab. Myself, along with a few others, are working on a project to develop a semi-automated approach to identify and monitor Phragmites using a combination of drones and satellites. Our work up until this point has been focused on the use of drones, so most of what I cover today will be specific to drones themselves. However, at the end, I will briefly touch on our future work with satellites. A big challenge that we have with Phragmites is the fact that we can't control it if we don't know where it is on the landscape. And due to the areas that it invades, that can be difficult to find with traditional in situ surveying methods. Wetlands, as we know, are difficult to survey already based on flooded conditions or inundated soils, making traversing them difficult. But on top of that, we also know that wetlands just aren't in our backyard or in our neighborhood park. They do exist in areas that are remote and difficult to access. And so the challenge becomes being able to survey these areas effectively and efficiently. And that's where remote sensing, and in particular drones, have this potential to be a valuable benefit to Phragmites management, specifically because they give us access to these areas that are not generally easily accessible. So if we think of the image that I showed you on the previous slide, there is really only a single access point to that wetland. With a drone, we can go to that access point, we can set it up, deploy it, and as long as the remote pilot remains in visible line of sight between the drone, or with the drone, we can collect imagery over these areas rather effectively and efficiently. So we're talking about surveying areas on the order of hundreds of acres in a matter of hours. And so what this means is that we can survey these areas much more quickly, resulting in much more frequent surveys. So we can survey these areas pre and post treatment to get an idea of how well treatment practices are working, which could ultimately drive management of Phragmites. On top of that, drones have a couple characteristics that are beneficial for identifying and monitoring Phragmites patches. First of which is a very high spatial resolution. So we're talking about spatial resolutions of 2 to 5 centimeters. And if you look at the image on the screen here, this is a drone image acquired over a patch of Phragmites in Duluth, Minnesota. If you look at the laser pointer, the patch of Phragmites is here in the center. However, at the edges, you can clearly see some of the fingers coming off the patch, where we can see where the patch is actively expanding, which is of course beneficial to know for monitoring these patches. On top of that, with the way that we collect drone imagery, we're able to create 3D models of the surface. And so instead of trying to identify features just using two-dimensional imagery, we can now incorporate the third dimension through the use of what's called a canopy height model, which is just an estimate of the height of vegetation within these areas. And so we truly do have a lot of information that we can use to identify and monitor Phragmites with this imagery. And it's great that we can sit here and talk about these potential benefits, but it doesn't really mean anything unless we test it. And that's specifically what we've done and what we're continuing to do. Today, I'm only going to be talking about 2 questions that we've been trying to answer here over the last year. First of which is being, can we identify invasive Phragmites only using the 3-band drone imagery in a semi-automated approach? So this is your red, green, blue spectral bands. And then the second question focuses on height. We know that height is a very important distinguishing characteristic when identifying Phragmites in the field. And so we wanted to see if including the height of vegetation through a canopy height model increased our ability to identify Phragmites. These 2 questions have been tested on 4 study sites, 3 of which are in Duluth, Minnesota in the St. Louis River estuary. On the top right, you have Hallett Dock, mostly dominated by forested wetland and shrub scrub wetland. Within the study site, there are no known populations of Phragmites, and so this area has been used to test to see what vegetation is being misidentified as Phragmites. The imagery that you'll see in subsequent slides was collected in 2018 in the first week of August. Grassy Point Park is the second study area. Some of you are well familiar with this location. It is mostly dominated by cattails. However, there are 3 patches of Phragmites present. The imagery that we used was acquired in 2017, so it looks a little bit different than it does today. And then Talus Island in the bottom left, similar to Hallett Dock, mostly dominated by forested wetland. However, there is emergent vegetation on the borders of the island and on the shoreline of the estuary. Similar to Hallett Dock as well is the fact that it doesn't have populations of Phragmites, and it's used to test for vegetation that's being misidentified. Talus Island was also acquired, and the imagery was acquired in 2018, the same time as Hallett Dock. The 4th study area is located in Saginaw Bay, Michigan. Saginaw Bay is an active area of Phragmites management, and I'm sure the next talk is going to go into this in quite a bit more depth. On the right, you see the study area. It's mostly dominated by brown. This is standing dead Phragmites. However, there are 5 patches of Phragmites actively growing during the time of the survey, which was conducted in August of 2018. And so to quickly point out where these patches of Phragmites are for reference, they're outlined in red in Grassy Point Park on the left. In the top left, you have a patch around the small linear basin. A patch is right here, but that is excluded from the study area. The second patch is at the end of this canal. It's interspersed with alder trees. And then the third patch is this large circular patch in the bottom center. Saginaw Bay on the right has these 5 patches that are in the bottom half of the study area. All of these are emergent and they're not growing on the shoreline. So to get into a little bit more of the nitty-gritty behind how we're doing this, we use a method called object-based image analysis. If you look at the picture on the right, this is an image over an airport in Rochester, Minnesota. We know that images are made up of a grid-like system of pixels, and if we were going to look at this image and try to identify something from it, we wouldn't just look at an individual pixel. What we'd really be doing is grouping pixels together and using the size, the shape, and the color of those groups of pixels to identify features. And that's exactly what object-based image analysis is, where we give the computer a more humanistic approach to identifying features. And so we create groups of pixels, which are shown on the right. This is done through a process called segmentation. You don't really need to remember that, just the fact that we group the pixels together. You can see them outlined in blue, and you can see they clearly represent real world features in the image. So the outlines of agricultural fields, the borders of buildings, and the edges of roads. And so we can then use the same methods that we use to identify features in this approach. So the computer can use the size, the shape, the color of these groups of pixels, as well as contextual information. So to give an idea of what that is, it's if you have a very small body of water right behind a house, we know it's likely going to be a pool and not a lake or an ocean. And in the context of Phragmites, if we have an already known patch of Phragmites, if there's some unknown vegetation within the middle of that patch, it's likely also going to be Phragmites based on how we know Phragmites to grow. And so this is a semi-automated approach where we build the step-by-step process that the computer follows, allowing us to include this contextual information and other expert knowledge. And then once the model is built, it's automated thereafter. And so what you're seeing on the slide right now is a simplified workflow of how our model works. To answer the first question on only using the red, green, blue drone imagery, so once again, 2-dimensional imagery, we begin with this unclassified image. We break the image up into groups of pixels. And the first thing that we do is separate everything that's vegetation from not vegetation. This is done using the optical imagery, so the colors within the image itself. And to identify Phragmites, we use texture. And so if we think of texture in front of us, our desk or a hardwood floor is going to be very smooth compared to something like carpet, which is going to be rough. We can calculate similar things in imagery based on the shadowing within the image. So something like vegetation will have a rough texture because you have shadowing between the stem and the leaf of an individual plant, as well as shadows being cast by the neighboring plants. Something like a road is going to have a smooth texture because there is no shadowing across the surface of the road. And so we can use textural algorithms to identify Phragmites from the vegetation. To answer the second question, including the height within the model, the only difference here is now the inclusion of that canopy height model. What this allows us to do is to split the vegetation into different height classes. So in this case, everything above 4 meters is considered a tree, everything below 2 meters is considered low vegetation, and everything between 2 and 4 meters is considered potential Phragmites. And then to identify the Phragmites further, we use the same textural algorithms. So to move into more of the interesting part, some of our preliminary results, we've found that only using the two-dimensional red, green, blue drone imagery is not enough to identify Phragmites from drone imagery. If you look at the figure on the screen, everything shown in bright green is Phragmites that's correctly identified. Everything that's shown in red is vegetation that's being misidentified as Phragmites. You can see in Grassy Point Park and Saginaw Bay, all of the Phragmites is correctly identified. However, when you look across all 4 study sites, you see that there's widespread misidentification. This means that a lot of the vegetation that is within these areas shares similar textural properties to Phragmites, and just using texture alone isn't going to be enough to identify it. So we find the Phragmites, but we can't differentiate it from anything else. However, once we include a canopy height model or include the estimate of vegetation height, we see a much different response. In the Duluth study site, specifically Grassy Point Park, we see a much cleaner classification or identification of Phragmites. 2 of the 3 patches are correctly identified. The extents of those 2 patches are not 100% perfect, but they're pretty close. They get a majority of the Phragmites. The patch at the end of the canal is missed. Once again, that's interspersed with alder trees. And so you can't actually see it in the drone imagery. And if we can't see it in the drone imagery, it's unlikely that the computer is going to see it or magically identify it. However, when you look around Grassy Point Park, there is very little misidentification. Pallet Dock and Talus Island are similar. They are much better than not using a canopy height model. You'll notice that there are areas that are being misidentified still. This is an active area of research for us, as we obviously want to reduce the amount of misidentified vegetation as much as possible. Things that are being misidentified are things like shrubs and some of your taller cattails that overlap in that bottom end of that height threshold. But overall, using canopy height model is important. Saginaw Bay is a little different. I included this to highlight some of the challenges that we have. Phragmites is only being correctly identified in 1 of the 5 patches. This is due to the quality of the canopy height model used in this study area, and so it severely underestimated the height of vegetation. And so what this means to us is that the UAS or the drone that was used in this situation might not be appropriate to estimating the height of emergent vegetation. And so this is another area of active research for us where we are looking to see what is actually needed, what technologies are needed for us to accurately estimate the height of vegetation and then subsequently identify Phragmites. And so the big takeaway of what we've found so far is that a canopy height model is critical to identifying Phragmites accurately, not because a canopy height model alone can identify Phragmites, but specifically because it allows us to remove a lot of the vegetation that's being misidentified as Phragmites. And so when we look on the figure here on the left, you see widespread misidentification without the use of a canopy height model. And then on the right, you see a much cleaner classification or identification of Phragmites because the canopy height model is included. And so this makes sense because Phragmites is structurally different than a lot of the other plants that are within this wetland. And so moving forward, one of the biggest challenges that we have for large-scale identification of Phragmites, knowing that canopy height models are critical to the accurate identification, is the fact that it's hard to get a canopy height model from the same time as the optical imagery is acquired. We do have LiDAR in Minnesota, but it's collected infrequently due to its cost. And so that leads to us trying to find another way to create these canopy height models. And there's another type of remote sensing data called commercial satellite stereo retrievals. basically the idea behind this is the way that the satellite imagery is collected, you can create surface models or 3D models of from the imagery based on how it's collected, so similar to the drone imagery. And looking at the screen here, this is just a collection of by one company. Everything shown in pink here is commercial satellite stereo retrievals. Unfortunately, I can't zoom in just to the Midwest or Minnesota, but you can see that there is a lot of imagery being collected. A lot of imagery is being collected over Minnesota. And so we believe that this is going to be our avenue for large scale identification, specifically because we are going to be able to have a canopy height model from the same day as the satellite imagery. So we won't have any discrepancy of changing distributions over years when trying to use a canopy height model from a couple of years ago. But like I said, this is still active research for us, and hopefully in a year or two, we'll have a little bit more to present on this. And so with that, I'd just like to make a few acknowledgements. MTRI and NRRI provided some of the drone imagery that you saw today. And then this project is being funded by the Minnesota Invasive Terrestrial Plant and Pest Center. And with that, I know I went over things pretty quickly, but if I'll take any questions, or if we don't have time to cover it now, feel free to send me an email.
We have plenty of time for questions, so about 5.5 minutes. And our first one for you is size, shape, and color enough to determine if a patch is native or invasive Phragmites?
So the big reason why we think Phragmites can be, or invasive Phragmites can be identified from other vegetation is because it's structurally different than a lot of native plants within wetlands. So it's very tall and it forms very dense monotypic stands. Native Phragmites doesn't necessarily do that. There are some unique cases where some stands are dense or have similar densities to non-native Phragmites, but to answer your question, no, I don't think that's enough.
The next question is, how can your process identify Phragmites patches that are less than 2 meters in height, which likely represent new invasions or mature patches early in the growing season?
Yeah, so that's also an area of research for us. We know that not every single patch of Phragmites is going to be above that threshold. And so that threshold is likely to change. I can't give you a good answer on how we would be doing that right now, but maybe in a year or two, I'll have an answer that works.
And then, since you're looking at both drone imagery and satellite imaging, what would be like the best circumstances for using drones over satellites? Is there a certain scale at which you would say it's better to use a satellite?
Yeah, so drones, I envision being more geared towards monitoring patches that are already identified. The areas that they can cover, a couple hundred acres in a matter of hours, which is still beneficial in areas that are remote and difficult to survey with traditional physical surveying. Drones will be able to survey those locations effectively and efficiently. Satellites are going to be more large-scale identification, so satellite images cover multiple square miles, and so we're talking, county level, state level, large-scale identification which is the goal. So, yeah, I think that answers that question.
All right, great. It doesn't look like we have any further questions from the audience, so thank you very much. And our last speaker today is Charlotte Weinstein, Mapping Invasive Phragmites Treatment Outcomes with UAS Imagery.
Great, thank you. Can you see my screen and hear me okay?
Yes, it can.
All right. So my name is Charlotte Weinstein. I'm a researcher at Michigan Tech Research Institute based in Ann Arbor. And today I'll be talking about our experience with mapping Phragmites, also with UAV imagery. First, I'd like to acknowledge my teammates listed on that slide. I'd also like to acknowledge that this particular effort was part of a larger project, mapping and monitoring of invasive Phragmites in the coastal Great Lakes using remote sensing data from multiple platforms. That was funded by the USGS Great Lakes Science Center. The PI of that project was Laura Bourgeot-Chavez, and it's also part of a larger team listed below. This project was completed in partnership with the Phragmites Adaptive Management Framework, also known as PAMF, which Sam introduced earlier. If you want to know more about this, I suggest that you will tune into our talk later today. But our role with this particular collaboration was to test the effectiveness of using a high-resolution drone image to incorporate that into adaptive management and monitoring efforts for Phragmites. We did this by visiting 20 sites distributed across Michigan, Wisconsin, and Ohio between July and September of 2018. All of these sites were enrolled in the PAMF program and their locations were provided to us by PAMF. And at each of these sites, we collected a combination of vegetation ground truth data, drone imagery, GPS points, and GPS tagged photos. And these data were used together in order to create interpreted maps with vegetation classifications. This is a rough overview of the methodology we used to create these maps, and I'll be diving into each of these steps with a little bit more detail in the following slides, starting with our data collection of both drone imagery and vegetation data. The drone imagery was processed into orthophotos. Some examples of what those look like are in the bottom left corner here. And then that data was then turned into some classified maps that were then used to evaluate Phragmites cover at these treatment sites. And some examples of what those look like are on the bottom right. The vegetation data that we collected was used to inform our classification and act as ground truth. This was very important for our classification step. On the right here is an example of what our field sheet looked like. The information that we collected included things like ecosystem type, species present and their distribution across the management unit, relative percent covers of different cover types, and some GPS tagged photos of the ground. An example of that is on the lower left-hand corner here. The UAV imagery was collected with one of two drones. At most of our sites, we use the DJI Mavic Pro. A picture of that is in the lower left-hand corner here. And then at two of our sites, we use the Phantom 3 Advanced, and some action shots of that are in the lower right-hand corner of this slide. The drones were flown in an automatic grid pattern to make sure that there was adequate overlap between the individual still images to make sure that they could be stitched together into an effective orthophoto. The camera resolution we used was 12 megapixels. And at two of these sites, we also tested use of a different camera that included a near infrared band. These are some examples of what those raw drone images looked like. On the left-hand side, we have an oblique photo, which just means that the photo was taken with the camera pointed at an angle. These are nice to get a general lay of the site, but weren't used in the classification. Instead, we used what we call nadir images, which are taken with the camera pointing directly down. And that's on the right-hand side of this slide. And you can see a pretty noticeable pattern with these Phragmites patches. All the emergent vegetation in this particular photo is Phragmites. Connor already mentioned that it has a distinctive mottled texture to it because of all the leaves and the seed heads. But you might also notice that it has a bluish green color to it, which was also useful for image interpretation. And this bright green stuff in the image is lemna or duckweed. Here's an example from a different site. Again, on the right-hand side, we have a nadir image, and this dark green vegetation is actually typha. You can distinguish it from Phragmites based on the darker color, and it also has a slightly different texture to it based on the structural differences from Phragmites. And then this brown vegetation towards the center of the image is all dead vegetation. Some of it, which was Phragmites, some of it was typha. Once it's dead, it turns brown and it becomes difficult to distinguish the two from each other. For data processing, we used a program called Agisoft Metashape, Below are some screenshots of the process we used to create our data layers from this imagery. The individual images were stitched together to create a merged composite image that was georeferenced to the site's location, also known as an orthophoto mosaic. And the pixel size of this orthophoto was 3 centimeters. We also used Agisoft to create some digital surface models to characterize the relative elevation in the study site. And those were had a resolution of about 13 centimeters. Like Connor explained just a minute ago, we also used a process called object-based image analysis to classify these images using a program called eCognition. The classifier algorithm we used was nearest neighbor, which is a built-in method in eCognition. And the types of inputs that we used included things like mean pixel value for each band as an approximation of color, the visible atmospherically resistant index, also known as VARI, which is sort of like NDVI, but instead of including an infrared band, it only uses the red, green, and blue bands, and also some relative geometric and positional information. And the way this process worked is demonstrated in the images below. On the top left, we have just the regular orthophoto before anything has been done to it. On the top right, we've segmented the images. Again, that means grouping the pixels together into distinct entities that can then be used for more detailed classification. It also helps save processing power so that instead of looking at every individual pixel at a time, you're now looking at aggregate groups of them. Once we had the image segmented, we drew training samples that were informed by our ground truth data and image interpretation. In this example here, the purple splotch is a training sample to train the classifier on what Phragmites looks like in this image. And that green splotch on the is a training sample of what Lemna looks like. And then once we had all that data set up, we ran it through the nearest neighbor classifier, and the type of output we got was something like the bottom right image below, where Phragmites has been color purple and Lemna has been color green. And again, the goal of this activity was to create a vegetation map that could be used to measure Phragmites cover to test whether that would be an effective use of quantifying treatment success. In order to judge whether this was a successful endeavor or not, we wanted to calculate the accuracy of our vegetation maps. For the purposes of this particular analysis, since our main category of interest is Phragmites, we aggregated our different classes into 4 distinct categories, those being live Phragmites, dead Phragmites, other vegetation like trees or shrubs, and other things that are not vegetation like open water or pavement. Within each of those 4 categories, we chose a statistically chosen number of validation points placed within each category. And at each of these points, we looked at how that point had been categorized by the classifier and whether that matched what we were seeing in the imagery. And using this method, we had an overall mean accuracy of 91.73%. And that's across all 4 classes across all 20 of our sites. Breaking down that accuracy result a little bit more, our mean and median overall accuracy were about 92% each. For live Phragmites across all of our sites, we had an average user's accuracy and producer's accuracy of about 90% each. For dead Phragmites, the accuracy readings were a little bit lower. Average user's accuracy was 76.5%, and average dead Phrag producer's accuracy was about 85%. Here's some examples of what those sites actually looked like from the air. And on the left-hand side of this slide are some examples of what the site looked like from the ground. So this particular site is an example of what we consider to be a good site that's suitable for this type of analysis. It's a relatively large management unit, a little bit over 2 acres. There's evidence of treatment. The brown area in the middle is fragments of dead Phragmites. And that's borne out in the classification here. The bright yellow area is classified as dead Phragmites. There's a little bit of typha in this image, colored in bright pink, and the rest of the vegetation is various shades of blue, and the road is gray. I should mention that all of these maps I'm showing to you now are final products that we shared with PAMF to be distributed to the landowners for these properties. And here's an example of a 4-panel map that shows the orthophoto, the digital surface model, and the final classification. And for each of these classifications, we calculate statistics within the management unit. In this map, the management unit is delineated by that black boundary. And that was provided to us by PAMF and was drawn by the landowners when they enrolled in the program. In this case, we didn't detect any live Phragmites in the management unit, only dead Phragmites and other types of vegetation. Here's another example of a relatively small site. Because we have such high resolution imagery, you can still resolve details even when the site is very small, although we don't necessarily recommend using drones for such a small site as this. In this case, you can see evidence of treatment, a lot of dead vegetation in this map, which is colored tan here. Also, a lot of cattails surrounding the management unit. And this is a site where the dead vegetation was a mix of both dead Phragmites and dead cattail. It's difficult to resolve those when the vegetation is no longer alive. So we group those together into a single dead vegetation class for the purpose of this particular product. Here's another site that's very interesting. It's on the beach. Phragmites was once present here, but it's been covered by sand over time. You can see in this classification, most of it is categorized as sand. The green areas are shrubs. But if you look at the history of this site using Google imagery, it tells you a little bit of the story behind this site. So starting from 1998, you can see the progression of this site. In 2005, this very clearly very dense vegetation, likely Phragmites. And then in the subsequent years, you can see the shoreline retreating. I really like this as an example of how historical imagery can really help you tell a story of the history of a site. This site is an example of one of the challenges with using drone imagery. The site did have a decent clump of live Phragmites, which you can see in the ground photo on the left here, but it was hidden under canopy cover, so we couldn't actually detect it using our drone. So in this classification, it was just categorized as trees because we couldn't penetrate through the canopy to see below. And again, because we couldn't see it, nothing was categorized as live or dead Phragmites. Here's one more example. This is one of the sites where we use near infrared imagery as well, which I'll show in the following slide. This is a case where we actually adjusted the original management unit that was provided to us, since the original was mostly under trees and didn't capture a lot of this Phragmites patch that is encompassed in the new management unit. There was no evidence of treatment at this site, so most of this patch was classified as live Phragmites in purple here. And this area ended up being classified as mostly live Phragmites with a little bit of other vegetation. So at 2 of our 20 sites, we also tested the use of near infrared imagery to see if that could help inform the classification and get better results. At these sites, we used a $300 MAP IR camera, which captures green, red, and near infrared imagery. For these sites, we performed the classification twice, once using just red, green, and blue imagery, and once with the near infrared band. And for both of these sites, we obtained similar accuracy results. Site 129, which is pictured here, had the exact same accuracy rating. And with this side-by-side comparison, you can kind of see why the classifications came out very similar to each other. For the second site, there was a little bit of an improvement with color infrared imagery, but not by a lot. It's also worth noting, though, that we only tested this at 2 of the sites, so the results here aren't necessarily conclusive. This is a comparison of what the second site looked like using red, green, blue imagery on the top panel versus color infrared imagery in the middle here, and then the digital surface model is on the bottom again. I'd like to finish up with some recommendations for how we think that this technology might be effectively used for adaptive management of Phragmites. We think it would be helpful if at least 2 image collections took place, once before any treatment takes place, so you have a baseline for your measurements to compare against. Then you would apply your treatments as needed, wait for them to take effect, and then fly again to evaluate whether that treatment was effective and compare it against the initial flight. And this could be repeated over time throughout the adaptive management cycle to track your progress. Finally, some considerations for those who might be thinking about using drones. One of the most important things to consider is the characteristic of your site. Based on our experience with this project, we think that drones are probably best suited to larger sites greater than 1 acre. That's where you're really taking advantage of the capability to cover a larger breadth of area in a shorter amount of time versus a field-based survey. It's important to take note of if you have any overhead cover. For example, if your Phragmites patch is obscured by a tree canopy, it's probably not a good candidate for this type of tool. The timeline for data collection is also good to think about. Preferred collection will be both before and after treatment, so you can compare changes over time. And during the collection, it's also helpful to collect ground truth data as well if you can. That really helps to clear up any ambiguities that might show up in the imagery. Airspace considerations are also important to take note of. At the beginning of the study, we had a much larger list of candidate sites, but we needed to remove several of them due to FAA regulations. For example, some were too close to airports. So that's something to keep in mind as well. And finally, there are some startup costs to this type of analysis. For example, acquiring equipment, learning how to use a drone, acquiring licenses for the data analysis software that you want to use, and obtaining proper training as needed. That's all I have now. So thanks everyone for your time and attention, and I'd be happy to take any questions.
Thanks. We've got about 3 minutes for questions. So the first one is, for each Phragmites site, do you have an idea of how much effort and time are committed to serving individual sites?
Yeah, that's a good question. I'd say there's 2 components to that question. The first one is the amount of time that it would take to actually travel to the site and collect the imagery. The data collection itself is not too time intensive. The drone makes it pretty quick, and most of these sites were fairly small. The amount of time it took to process the image varied from site to site, which makes sense. A lot of it depended on how complicated the site was. For example, some more heavily invaded sites would have denser monocultures of Phragmites that are easier to identify. And so those would take less time to distinguish from surrounding vegetation. So I guess the short answer to that is it really depends.
And I have a question for both you and Connor, and it relates to how can practitioners use your tools in the future? Are there ways that they can download part of your code or use some of the classification data that you already have? And that way it makes it easier for them to process their own imagery in the future?
Yeah, that's a really good question. I don't know too much about open source with respect to drone imagery. Maybe Connor can speak to that a little bit. I would say that we can definitely share our workflows. We have a manuscript in progress right now that documents what we've done for this particular project. It's a little bit more difficult to share training data, not because we wouldn't want to share it, but because a lot of times the training data will vary a lot depending on the individual image itself. A lot of these drone images were taken at different times of day under different weather conditions, which influences a lot the individual pixel values. And so that's one of the challenges with this type of analysis.
Yeah, kind of just to reiterate what Charlotte said, sharing of e-cognition rule sets are difficult because you can't actively look at them unless you pay to use e-cognition, which I think is like a $10,000 license. There are open source methods for processing drone imagery. I don't remember if Agisoft has an open source option. I know you can pay for it. And then classifications done after processing or stitching the images together. There are multitude of open source options. One that we use is Orfeo Toolbox, which is part of QGIS, which is an open source GIS platform. As I guess for sharing what we did, right now, I don't have any intent to do that, at least the rule set, because it's still actively under development. But hopefully in the next few months, we'll have a publication out that details the workflow in more depth than I went into.
All right, great. Thanks to all of our speakers for their information this morning. And we've got our next session, which is going to be at 10:20. It's our second Phragmites session of the day. So thanks again for everyone's attendance. If you've got any specific questions for any of our presenters, please send them an email. Thank you.